The Bioscience Growth Council is a CBIA committee of biopharma companies, startup biotechs, and research institutions like Jackson Labs. Just some webinar details as we get started. Please use the Q&A function located in the middle of your screen to submit your feedback and submit any questions you might have. Also, the session is being recorded and will be posted at cbia.com. Now, the coronavirus has certainly upended our lives, personally and how we conduct business. So it's great to be here today because we're going to learn how to fight back against the coronavirus. Of course, the Holy Grail is a vaccine. Actually, that's not right. A vaccine isn't a Holy Grail. It's something mythological. A vaccine is something very real, quite findable and doable. It's just that creating a vaccine takes a while. Now, a vaccine for the novel coronavirus will probably be developed faster than any vaccine literally in human history. We may have a workable, safe, and effective vaccine in small amounts, small quantities, sometime this fall. But a widely available vaccine is probably at best something like a year away. That's why testing people for COVID-19 and tracking and tracing people who come in contact with people infected with the novel coronavirus is so important. It's critical too to go on with our lives. That's why it's important to understand how we can make our workplaces and factory floors safe for our colleagues, safe for our employees, and for business to be conducted. So we've got a great panel today to address all three of those issues. Lewin is an expert on vaccine R&D. He holds a PhD and an MBA. He's vice president of the biopharmaceutical company Sanofi Pasteur and heads Sanofi's BARDA office and new venture engagement. BARDA stands for the federal government's Biomedical Advanced Research and Development Authority. BARDA is in the thick of identifying and funding promising COVID-19 therapies. Charles Lee is our expert on COVID-19 testing and tracking and tracing. He's scientific director and professor at the Jackson Laboratory of Genomic Research. He's an expert on the human genome and how genomic variation contributes to disease. If you're wondering what the other designation after his PhD stands for, FACNG, it's Fellow of the American College of Medical Genetics, which is a big deal. To explain how to use all the information and deal with all the COVID-19 issues coming at us in our businesses, Brian Montanari and Boedicker from HABCO are with us. HABCO is an aerospace company that makes all sorts of high-tech aerospace components for commercial and military applications. Brian is president and CEO of HABCO, and Carol is HABCO's HR VP. HABCO has been a tremendous leader in showing us how to create and maintain safe workplaces in the shadow of COVID-19. So I think it makes sense to go from what I consider the macro, what is the coronavirus and COVID-19, and how we deal with it through testing and in our workplaces. So I'd like to start with Clem. Clem, I'd like you to talk a bit more about what you do at Sanofi and how your work focus has changed in the wake of the novel coronavirus.
Thank you, Paul. Good afternoon, everybody, and thanks to the CBI for the opportunity to speak to this group. It's always, I'm looking forward to hearing and learning from the other speakers. So how has my life, Sanofi's life changed as a vaccine company? My role within the company was to deal with BARDA, which is a Biomedical Advanced Research Development Agency, which is part of HHS. And most of my time in that role was spent on pandemic influenza preparedness. So BARDA is one of the leaders in pandemic influenza preparedness and works very hard to prepare for an influenza pandemic as opposed to seasonal influenza, which we see every year. The influenza pandemic would be if a novel strain of influenza emerged. I also dealt a lot with policy issues related to vaccines. Life dramatically changed for me and many colleagues at the end of January, early February, with the emergence of SARS-CoV-2. We are focusing on developing a vaccine. Our partnership with BARDA on pandemic influenza helps. The difference here is for pandemic influenza, it's a novel influenza strain, but you can use the technology that you have for a seasonal vaccine to develop a pandemic vaccine. You make a new seasonal vaccine every year, so you change the strains out, and you do the same essentially for pandemic, but we know what the vaccine looks like. Here, the company and other companies and NIH, now a bunch of other people are working to develop a target vaccine for scratch against the coronavirus. Most of us are working on targeting the spike protein, which is on the outside of the virus, because that seems the most promising candidate. And while there's been some work on SARS is also a coronavirus, we're really starting from scratch. So figuring out how to produce the vaccine, test it in non-clinical testing, then get into small-scale testing to just show that the vaccine demonstrates an immune response. That means it could protect you. And then moving towards efficacy studies to actually show the vaccine works. So for the last 3 or 4 months, my life has been consumed by moving the product forward. And many of my colleagues, we have a site in Meriden, Connecticut that's actually producing the vaccine candidate for the technology that we're using. But I think there are probably about 120 people involved globally within Sanofi on this program at the moment.
Yeah. So let's just back up for a second. And so what exactly is a vaccine compared to, say, a drug? So a vaccine, I guess, is to prevent disease versus a drug which treats it. So
that's spot on. A vaccine is usually to prevent disease. So, and as I think it was Ben Franklin said, an ounce of prevention is worth a pound of cure. It's one of the great public health interventions after clean water in terms of reducing disease. So what a vaccine does is it mimics an infection without giving you the symptoms, and it produces, you produce an immune response, and then if you're infected again, the immune response kicks in and protects you from the infection. There are different mechanisms for how it works, but that's the general principle. And one of the reasons people are encouraged about finding a SARS vaccine is if you survive the infection, that means that the immune response can protect you. And that's what we're seeing here. People are recovering. So if you can recover from the disease, we should be able to develop a vaccine that gives you that immune response so that you never actually get the disease.
I mean, if there were a vaccine, could you treat and somebody came down with COVID-19, would a vaccine work as a treatment?
In this case, probably not, because of the speed at which the vaccine you develop an immune response that you would have to have a very, very quick immune response. A vaccine you'll need to be given prior to exposure to the virus.
Got it. So as I said in my introductory comments, we're dealing with the novel coronavirus that upended our lives. What's novel about it? What's new about this virus?
That's a great question. I think we have seen, first of all, there are coronaviruses that are human coronaviruses that cause infection in humans. I think there are 4 or 5 of them. The difference here, we've seen coronaviruses emerge from animals. They come from bats, and then they go through either camels or other species and into humans. We've seen the SARS outbreak, and we also see these outbreaks of MERS, Middle Eastern Respiratory Syndrome. Both are caused by different coronaviruses. The difference here, and this is what turns it into a pandemic, is that you have something that transmits easily in humans. So it transmits from human to human relatively easily, and it's transmitted It's transmitted now globally, so it's become a pandemic. The other difference between, let's say, SARS and SARS-CoV-2, this COVID-19, is it's you have asymptomatic transmission, or people have mild symptoms, so it's very difficult to track. I think Charles Lee will be talking about it, and Brian and Carol, or the challenges that come. But that's really the difference. Is it's unlike SARS, where people got really sick. This is less. It's less easy to track. So think of Ebola. It's pretty easy to know who has Ebola. Here, we don't really know necessarily who's carrying it and then who can transmit it.
Got it. So before we talk more about vaccines, all the different vaccine research that's ongoing, just why are viruses so difficult to treat? So a bacterial infection, we would hope that a antibiotic would be developed to treat the disease once you get it. Why are we so focused on doing something before you get the disease? Because the virus is so difficult to treat once you have it. Why are viruses so complicated? Because they're simple little things, right?
I think for vaccines or for vaccines, you have bacterial and viral vaccines. So you would want to treat either one. Bacterial infections are becoming more difficult to treat due to resistance. But I think the reason viruses are difficult to work with is that they are essentially blueprints looking for factories. So they infect human cells. So by definition, to kill the virus, you're usually trying to kill the cell as well, unless you can find a drug that stops the virus from entering the cell. So it's just a more complicated thing to deal with.
Sure. So that makes that, because of that fact of viruses, it makes finding a vaccine very, very important. So I think there are more than 100 vaccines in development. And so if you could categorize them for us, I think there are 4 or 5 different types of vaccines. Some people talk about traditional vaccines versus new, and certainly you guys are on the forefront of kind of a new technology.
So there are the U.S., globally, the oldest in the U.S. there's 4 technologies. There's another technology that's not being looked at as much in the U.S., which is an inactivated virus vaccine. So basically, you kill the virus, inject it into people, and they mount an immune response. What the approaches that people are using here are 1 or 2 of them are what would be called vectored vaccines. So basically, you take a virus that you can put the gene, in this case, coding for the spike protein. Everybody's looking for spike protein. And you put it into people, and they mount an immune response to that. That's one approach. The second approach is mRNA. Many of you have probably heard of a company called Moderna. That approach is essentially you inject mRNA, which is the blueprint for the spike protein, into the individual. They make the protein in their cells, because the mRNA is wrapped in a little lipid nanoparticle. Think of it as a gumball, clear plastic gumball with the mRNA, and it fuses with the cell, and the cell then is taken over, and you make the spike protein, and your body mounts the immune response. So it's like your body produces things. This is a new technology. It's never been shown to work or licensed in a vaccine, but it's extremely promising. The other technology is that we're working on is what's known as recombinant DNA. We already produced a flu vaccine based on that, which is what you do is you take the gene of interest, you put it into a virus to infect cells, and then you harvest the protein, again, the spike protein. And here we combine it with an adjuvant, which is something that boosts the immune response. So it makes you get a better immune response. And here you're taking the protein and injecting it into humans. So those are the 3 or 4 approaches. Some are more new than others, but they all are basically the same idea of targeting an immune response against the spike protein.
So it's part of the issue with any vaccine for the novel coronavirus, the manufacturing capacity, and so your vaccine the direction you guys are taking, in some sense, is a faster way to we'll end up with a quicker way to manufacture.
I think all the approaches are different, and what you're bringing out is something I should have mentioned, is in this case, it's not enough to show the vaccine is safe and effective. We need very large quantities to be able to vaccinate about 300 million Americans and billions of people globally to end the pandemic. So the ability to scale up and manufacture these large amounts of doses is critical. The technology that we use, we already have experience in manufacturing. Other technologies may need to be scaled up, or the experience in manufacturing large quantities may be less, but that's a key component. And what you're seeing through Operation Warp Speed that the US government has started is they're working on scaling up manufacturing in parallel to development. So everything, normally all these things are done sequentially. You might identify your vaccine candidate, you would then optimize it, put it into man. If it looks good, you move forward, and you would then maybe start manufacturing once you're pretty confident that you have a product. Here, everything is being done in parallel, at risk, not at risk to the patient, but risking resources, both the people and the funding, to do this, so that we can hopefully by the end of the year or early January have a lot of doses available if we show one of the vaccines works.
Right, so I think a couple of biopharma companies, including Moderna, is starting manufacturing during the phase 3 trial that's supposed to begin in July. I mean, so what if that trial is unsuccessful? What happens to that vaccine?
It won't be used. So that's the risk. When we talk about manufacturing at risk, people are manufacturing. Normally, you'd wait to see whether you had the results to do that, but that could delay the availability of a vaccine by, let's say, 6 months. In a pandemic, you're going to hear from the other panelists how important it is to have a vaccine, I believe, to end the pandemic so we can get back to life at normal. So saving 6 months, given the economic cost of the pandemic, makes sense. It's, for the amount of money that this is costing the economy, our strategy, by ours, I mean the government strategy, is to produce the vaccines at risk in case, so that if they work, they are available.
So you're in partnership with GSK. So where will you be manufacturing your vaccine?
We will be manufacturing the drug substance, the protein, and in Pearl River, New York, and potentially other sites. Obviously, our team in Meriden, Connecticut, will contribute to that. And then GSK is manufacturing the adjuvant at their sites, some of which are in the US.
Well, thanks so much, Clem. We'll probably come back to you too, but let's talk a little bit about testing. So Charles, I'd love to, first, I'm always interested in people's lives, especially the science front, your lives before COVID-19, and then how your research focus has changed. So just tell us a little bit about sort of your life before COVID-19, and then how it's kind of switched very dramatically with COVID-19.
Thank you very much. So here at the Jackson Laboratory, we are a nonprofit research institute. We have approximately 30 faculty members doing research in the areas of immunology, the microbiome, cancer, human genetics, and we have, the projects we're working on. In many ways, the focus of the Jackson Lab for Genomic Medicine is to come up with new diagnostics, new therapeutics, new genomic solutions for human diseases. As you can imagine, when the COVID-19 pandemic struck, a lot of the researchers, including myself, certainly started to gain a lot of interest into the biology of COVID-19 and how can we use our respective training and expertise to complement one another to learn more about the disease. Certainly, the immunologists were all over this initially, but even those that were working in genomics were, of course, very interested. Everything from being able to sequence the virus, to look at the mutations that are occurring in the virus, to use that as a tracer, to see even what we call molecular tracing, how the virus is moving around the country, and what changes that are in the virus that would alter its biology as well. So I think, like a lot of the academic institutions, what we found is the scientists here at the Jackson Laboratory for Genomic Medicine have really been focusing in on learning everything we can about the virus and sharing that information to other scientists around the world so that we can collectively tackle this problem in an expedited manner.
Sure, just to back up a second, when you talk about sequencing the virus, so what is just explain to our audience, understand exactly what does that mean? Everyone talks about that. That's so important today in biomedical research, sequencing.
So basically, the virus is a single-stranded RNA molecule. That's the genetic makeup of this particular virus. And what we and many others actually want to do is to actually find out the exact nucleotide sequence of the virus to find out if there are any changes or mutations that could reflect changes in the biology and infectivity or even just as I mentioned earlier about being able to trace. It's like the different changes that occur over time sort of give us a fingerprint of the viruses. And so we, by analyzing, for example, the viruses that come into our diagnostic lab here at the Jackson Laboratory, when we actually can sequence from the virus from one individual to another, that are in one individual to another, we see that the actual viral sequence is actually not the same, not exactly the same. There's small changes that we could see from the virus in one person to the virus in another person. And so earlier, people were wondering if, for example, changes in the virus genome could explain the variety of symptoms that we're seeing with the COVID-19 pandemic. So as you know, some people can get infected with COVID-19 and they actually experience little or no symptoms. Others have very severe symptoms. It's thought that some of this is probably due to differences in our own genetic makeup in our own bodies and differences in our immune systems, etc. But we couldn't rule out the fact that maybe there's also changes in the virus itself. Right now, I don't know if, as many people are actually sequencing the viruses, we're seeing a lot of mutations. That's to be expected. I'm not sure if we're seeing a lot of evidence for the fact that those changes are directly correlated with whether the virus is more infectable or causes more severe symptoms. I know that there was a study that just came out literally in this past week from the Scripps Institute, where they suggested that they've seen some mutations occur in the virus, which may increase the number of spike proteins that are on the virus. And in vitro studies, in cell cultures, it looks like it's it's able to infect those cells easier. And so they're speculating whether or not that could mean that there's a mutant strain that is more infectable. I think it's quite early to make those conclusions yet, but that sort of gives you a feeling for some of the data that can arise from actually figuring out the exact nucleotide sequence of the virus.
Sure, that's very interesting. So again, just so I understand it, the sort of the inflammatory response that some people get with COVID-19 versus the very mild symptoms others get, and I think there's some cardiovascular separate disease profiles. I guess otherwise people have been saying that's more to do with the genetic profile of the patient versus the virus.
Exactly, that's correct. I think there's a lot more of what we call the host genetics differences that would contribute to that variability than the viral. But like any scientific problem, you really need to do it properly, comprehensively, you need to look at it from all angles, right? And not just assume one thing. So getting as much information from both the host as well as the virus is important.
Yeah. So just quickly, just one of my questions. I was going to end with this, but just it's certainly going across social media. Has the virus mutated? There's some like it's become a milder disease, as viruses do. Is that correct as well? Usually over time they become.
Right. Yeah, there's no doubt that the virus is mutating. That's I think all geneticists know that. Whether or not the virus is mutating into a less severe form, I think that's it's too early to suggest that. I haven't seen data that would convince me that that's the case.
Yeah. So let's talk about testing. So if you could just tell us the different types of testing for ongoing disease, and also maybe touch on antibody testing. But there are several different types of testing for ongoing disease, and some with different levels of reliability. So
absolutely. So in two general buckets, we can refer to testing as testing that is trying to detect the virus through the virus RNA or virus antigens. So you're just trying to detect the virus in the individual. And then a second class would be more of the antibody, what you're referring to as the antibody testings. Do you detect antibodies in an individual to the virus? So with respect to the first pool, most of the tests that are done for that is really just looking for the presence of the virus particles that contain the SARS-CoV-2 RNA. And so that's where you have, for example, the nasopharyngeal swabs or saliva or anterior nair swabs that collect mucosal specimens from the individuals. That is sent to a laboratory in general. And those virus particles if they're present, are cracked open, and then a test is looked at for detecting whether or not the specific RNA, viral RNA, is present or not. You can also
identifying the virus.
Exactly, the presence.
Pieces of the virus. Yeah.
And you can also accomplish the same thing through something called an antigen-based test. So rather than looking for the actual RNA particles or sequence in the samples, you're looking for particles from the virus itself, so the protein particles. And I would say the vast majority of tests that are out there to detect the virus are those that are detecting the RNA. I think of maybe over 50 of the tests that have been approved by the FDA for detecting the RNA of the virus. There may be about 3 or 4 now that are able to detect the antigens or the proteins of the virus. So the vast majority are RNA detection. Now, because of the way this pandemic has come through the US very quickly, and as you can imagine, the FDA has expedited the availability of what we refer to as EUA, emergency use access tests. And what we're finding is that some tests are a little bit more sensitive than others. So for example, some of the point of care tests that take a swab and then they call it these rapid tests, 10, 15 minute tests, for a variety of reasons seem to have a little bit more difficulty in their sensitivity than the more classical PCR-based tests. But they all accomplish the same thing, is do they detect the virus or not? With respect to the antibodies, I think the most important thing to learn about the antibodies is that, as we know, generally when we are exposed to the virus, our body develops antibodies to it. And so the conception is that if you have antibodies to the virus, you might have some protective immunity to that virus for reinfection. So if you had it once, if you've had the COVID-19 once, then you're less likely to get it again because you have antibodies that can, as Clement mentioned, can be used to mount an immune response faster or symptoms appear. And so the problem right now with the antibody test, though, is it's really the interpretation. I think people are very anxious to have an antibody test because they want to know whether or not, one, that they've been infected, and two, whether they're immune to subsequent infections. And I think it's really important to caution people against that because we, at this time, we do believe that there's some level of immunity, but we don't understand how much immunity, for how long, how high your titers need to be, et cetera. So, and combined with the fact that there are many studies around the world that have showed hundreds of individuals, cohorts of hundreds of individuals where they've been infected once, they developed antibodies, and then they actually get reinfected again. So that is why the WHO, rightfully so, has said that having an antibody test does not provide information on whether or not you're immune to the virus.
So what do we need to learn about the virus to figure out if the antibodies give you that kind of long-lasting protection?
So there's something called a neutralizing assay, where essentially it's an assay that will just determine if you take a serum from an individual with all the antibodies present and combine it with cells and combine it with the virus. There's deviations of this assay, but essentially when you take serum from an individual with all the antibodies, you take human cells and you take the virus, combine them together. Does that serum, the serum from that individual, inhibit the virus from infecting those cells? And so to really understand whether or not there's that level of immunity, I think that's really, those are kind of assays that we need to be moving towards. Until we understand that in order to get certain amount of immunity, you need a certain level of IgG antibodies. You may need to have some profile of your own immune cells and see that whole spectrum. But that's sort of where we're at right now in the testing phase for antibodies.
Mm-hmm. So I had to ask you too about, so Jackson Labs is very well known for its mouse technology, the use of specifically engineered mice to test things. Is that being used in the COVID-19?
Yes, absolutely. So our colleagues up in Bar Harbor, Maine, so the headquarters for the Jackson Laboratory are in Maine. It's a beautiful place to go to. If you haven't been there, you really should go and take a moment to enjoy the scenery, and then of course come and visit us at Jack's there. But what we've done is, as the pandemic started, we actually had in our repository a genetically modified mouse that has the human ACE2 receptor. And the ACE2 receptor is the major receptor on our cells that allows the virus to attach and then to be internalized. And so
it's a mouse. Its lungs are acting like a human lung to some degree.
Yes. So if you looked at the lungs, for example, they're behaving more like the human lungs with respect to the ability for the SARS-CoV-2 virus to attach and to internalize. And so they've actually been mass producing these mice and providing them to researchers around the world, particularly with respect to vaccine development. So I feel bad with Dr. Lewin here. If I say something wrong,
we're actually using the mice, so I know.
Okay. So when you're developing vaccine, some of the things that you do is you do what we refer to as in vitro studies, looking at cell cultures, et cetera. But ultimately, you want to do what we refer to as in vivo studies. And that one way is to look at how does the virus affect mice. And so the idea here is, for example, if I made it simplistic, if you had 5 mice with the ACE2 inhibitor, and you give 5 mice the vaccine, 5 mice without the vaccine, and then give them all the virus as well, presumably those that had the vaccine, if the vaccine is working, shouldn't develop symptoms of the disease.
So let's talk a bit about track and tracing technology, which is sort of a component of the testing. Mm-hmm. And just so if you could tell us a little bit of what that means and how it works.
Sure. So the contact tracing is incredibly important because basically anyone that's identified as positive for the SARS-CoV-2 virus means that they're actively shedding the virus and able to infect others. And so what contact tracing basically does is when we identify those that are positive, we quickly identify who individuals that were in close contact with the infected person over the past 24, 48 hours, etc. And then we want to get those individuals either isolated or tested themselves. And so that what it does is helps identify and contain individuals that have been infected, those that have potentially infected others. As you can imagine, in order for this to be effective, you need to get the results for the test pretty quickly. So our recommendations when we were on the open CT task force was that 24, 36 hours really need to get those test results. You can imagine if you don't get the test results for 4 or 5 days, effectively how difficult is it to identify all the people that you've been in close contact with for the last 4 to 5 days. And then you also have to find out who they were in contact with because the virus has had time to circulate. So it's really important to identify those people quickly and then isolate or get those people tested as well. So the state is actually has teamed up for this with Microsoft. There's a program called Contact CT, which you can learn more about on the web. And hundreds of government employees are working on Contact CT. And in fact, what's really nice is that they've also been recruiting hundreds of volunteers, university students that are trained appropriately, but can also help with the contact tracing. And this is a very important part of containing the pandemic.
Well, thank you very much. I think we'll hopefully come back to that too. And now I want to turn to HABCO and see what's going on, in the workplace and the very practical things that are going on. I mean, I'm amazed. I mean, just running an aerospace business generally before COVID-19 was complicated and difficult with all sorts of customers, and you want to keep those customers happy. So again, I'm really interested to sort of life before COVID-19 and how you're dealing with your employees, both in office spaces and especially on the factory floor, but then also meeting all your contractual commitments with your customers and meeting them on time. So
yeah,
I'll let you two just.
No problem. Thanks for having us on, Paul. Yeah, to say that the last 3.5 months has been a challenge would be a complete understatement. 3.5, 4 months ago, we were loving the commercial aerospace business, and there was a lot of effort being put into ramping up to hit that demand. And this is one of those times I'm thankful that we're pretty diversified between the government side of the house as well as the commercial aerospace side of the house. So now it's been a focus on how do we convert those resources and put them on the military side, because the commercial business has been affected quite a bit. But for us, we started very early on. We have a partnership. One of our locations is in Europe. So for us, we started seeing what was happening early on. So we wanted to jump on it as quickly as we could. And we started the focus in February, meeting as an executive team. And at the end of February, what we decided was we put 3 guiding principles together and will not allow ourselves to waver from those 3 things. And that's been one of the biggest benefits for us. The first and foremost is the health and safety of our employees, our suppliers, and our customers. Number 2 was providing our employees the flexibility they need, because now they are daycare providers, teachers, home shoppers for their family members. And then third, do that without compromising customer demand. And those 3 things come in conflict every day. And that's where our challenge has been. And how do we, I think we started off pretty well being on the forefront of protecting the employees. But as we learn more, how do we continue to adapt, and how do we continue to change our protocols to make sure that those three things are always at the forefront for us?
So how did you process, where were you collecting the information? I mean, who were you getting the sort of best practices from, and how did you process that as a company to come up with policies?
Yeah, initially it was just a lot of stuff on the internet, and then each of us on the executive team, we'd be doing research. And to say that we got addicted to this virus early on is really the reality to it. Carol, the amount of research she's done, and the rest of us, just whether it's internet, press conferences, talking to coworkers, talking to relationships that we have over in Europe, where they started feeling the spike before us. And it was just real time every day. Started off with us having a task force meeting every day at 9:30, and that started off 7 days a week, and now we're doing that 5 days a week. But we're continuing to keep that beat, and just whatever information we have, bring it to the table. Let's make a decision and go from there.
Sure. So is there anything, so now that we're several months into this, is there anything, when you look back, is there anything you wish you had done differently?
Yeah, it's a great question. Initially when we started, it was the basic hand washing and just education, and then we quickly move forward. I think really where some of the areas that we talk about quite a bit was probably a little bit further into the pandemic, especially as we started being able to do testing of our employees. We started doing saliva testing. We're the first non-healthcare company in the country to start testing self-administered tests on site. And we were really conflicted with that early on, because every day we're hearing the governor talk about need more testing, the president talk about need more testing, and here we are with testing available, and this essential business, but non-essential from a healthcare, from a frontline perspective. We're sitting there able to test. And initially we held off. I made a lot of phone calls to other folks to get this test in other areas. But once we found out there was plenty of capacity with this the company we were using, that every day that went by that they weren't testing, they were losing capacity. So we definitely had a lot of discussions about that, and we delayed some of the testing that we probably could have started a little bit earlier. Mm-hmm. And I think the other area was, when I say we wouldn't waver on any of those principles, I think we stuck to it very well. But we kind of looked back and said, okay, we developed processes and protocols to allow our customers to come on site and inspect product before we ship them. We have government inspectors that come on site and customers come on. But yet we wouldn't let some of our employees who really wanted to come back in the facility and grab something, we wouldn't let them come on site. And so we kind of looked at in the rearview mirror afterwards and said, okay, well, we can treat our employees who we've forced to work from home. If they need to come on site, we can treat them like a supplier and have them go through the same protocols that we have a supplier or a customer go through.
That's interesting. Yeah. I mean, you guys were one of the first to use the saliva test, I believe. So, tell us a little bit about what that is, and how did you come to learn about it?
Yeah. So I learned about it on LinkedIn. I have a contact of mine on LinkedIn who I knew personally through our daughter's hockey team. Didn't quite know what he did for work, and I saw him put a post on LinkedIn on a Friday that said his company is going for authorization under the Emergency Use Act. And it kind of piqued my interest. And that Sunday, he posted again saying that they received authorization. And so I immediately called him and said, okay, can I buy some? And he said, well, you really can't. You need to be a healthcare provider. You got to be, you got to keep in mind HIPAA, and you've got to comply with CDC and all these things. So I challenged him and I said, okay, Dom, how can we figure out a way to get industry essential employers to be able to buy this? So we basically worked around the clock that Sunday through Tuesday, worked with our attorneys. And then by that Tuesday, we developed a process that worked in conjunction with a third-party nurse practitioner. And Carol does all the administration of it. So I'll let her talk a little bit about it and how we've been using it.
Yeah. So how do you as a practical matter test employees? You do it on site.
Yes, we are able to do it on site. There's actually a pre-authorization process. So we developed a relationship with a nurse practitioner who handled all of the front end part of the pre-authorization. We also took it upon ourselves to work with our own attorneys to come up with our release document for our employees to make sure that we were protected on our end, as well as then the relying on the nurse practitioner externally. But essentially, any of our employees, if we were bringing somebody in-house who had been an external employee, they would actually drive to our parking lot, and we would arrange to actually have them complete the test in the privacy of their own vehicle. It's not invasive at all. It doesn't require any assistance. So we would literally take a test kit out. They would complete the test. We ship it to the lab, and within 24 to 48 hours, we have results. For our employees who are on site, the direct labor workforce, we met with them, we walked them through the entire process, because the whole communication piece, this is something unknown to so many people. So a lot of it was communicating what the process entailed. And once again, they were able to complete those tests in the privacy of their own vehicle. We shipped them out. Everything is kept confidential so that we protect all of the HIPAA related items. And so it's just been a really streamlined process for us. The nurse practitioner, it pretty much is just a secondary external person who's helping with the initial setup to make sure that we're able to administer a test to them.
Sure. So you have both office workers and essentially a factory workplace. So how does that work in terms of testing, and what are the different requirements?
So for us, although we're an essential employer, we made the decision up front that we were going to do as much as we could to provide the social distancing internally. So we actually mandated back in, the mid part of March that all of our non-production personnel would be actually working remotely. And so since March 16th, they have been off-site. So that was more of the technical challenge of getting them set up off-site. The remaining 50% of our workforce is our direct labor workforce. Those individuals stayed on site, and we split into 2 separate shifts to provide even greater social distancing with all of the standard protocols with the PPE. We actually have been doing temperature checks since the beginning of March. So we had a lot of those protocols in place. When we were able to secure the COVID testing, it just added another level for us. In the event that we had an individual, for instance, if they tested above the 100.4 with their temperature reading, we could immediately administer a test to them. And so, for the folks that are on site, it's been a lot easier logistically. For our remote staff, we coordinate with them externally so that they take care of sending in all the pre-authorization paperwork directly to the nurse practitioner. We schedule a time for them to come on site to actually then take the test, and then they wait the 24 to 48 hours for their results. But overall, it's been a very streamlined process. I've been in HR for 30 years. I never thought I'd see a day where I'm distributing specimen jars and giving people biohazard bags. But when you're faced with a challenge like this, you step up. And so this is a new thing I can put on my resume, I guess.
You don't need to update your resume, Carol.
So the 2 shifts for the factory. So something you had not done before. So I mean, that was a logistical huge business thing to do, mechanically, just to run 2 shifts. And it's done to meet the social distancing and meeting your customers' needs. That was the only way to do it. I mean, sort of like, restaurants are 50%. They're only going to allow 50% of customers in. They have to have double the time or double their prices or something like that.
Absolutely. I mean, we had always talked about and had a plan for a second shift. This just kind of facilitated it sooner rather than we anticipated. But I think, one of the keys was recognizing when we moved to the second shift, not only were we trying to make sure that we were providing a safe workspace for our employees and continuing to meet our customer commitments, but we also had to recognize that we were asking a lot of our employees. We were asking our individuals who had traditionally been first shift employees to now start working originally what was a 3:30 to 12 o'clock shift. We then had to migrate to 4:30 to 1. So it became, a challenge for some. And we said from the beginning, anyone who had concerns, please step up, let us know, let's have a conversation about it. But we set the parameters. We've continued to communicate to our employees throughout the pandemic, and just doing that split to 2 shifts, creating the social distancing, has just really been instrumental for us. It's just been another precautionary piece that's really helped us and helped to ensure the safety of our employees. It also protected us in the event that we had someone test on either shift. We could immediately then isolate that shift, but still, have a second shift of employees that could still maintain the operations. So, there were a lot of those different protocols in place that gave us some flexibility.
Yeah, and if I can add, Carol even went one step further than that and actually called past employees who left on good standing, who may or may not be furloughed at the time or didn't know what their hours of employment were, to have a list of a bench just in case we did have an outbreak on one of the shifts. We had a whole other crew that we could have brought right in and slipped right in with ease.
Yeah.
So have you had any pushback from employees who, despite all the procedures you put in place, the protections you put in place, who said, just nevertheless, I'm not willing to take this risk?
Yeah, it's
especially people who you need to have come into your workplace, who can't.
Yeah, that's been a very interesting element of this for us. I think culturally we started early enough where as we introduced temperature checks, they understood why. And then we started doing masks and gloves. And every day, so on March 23rd, I started a YouTube channel just to be able to every day provide a video to the entire company. Because I love to be able to just walk in the cafeteria and see everybody. We have monthly all hands meetings, daily Gemba walks where I got to see everybody. And now I can only see 1/3 of the company because I can't even be here past shift change on second shift. And I wasn't seeing the folks off site. So I started with a YouTube channel and every day a video was put out. And not just about what we're doing, but what we're hearing from all of the other resources that we're participating in. And that was a big element to us, so that by the time that the testing came, we had done enough discussion with it. Now, initially it was voluntary. So we weren't in a position where we can mandate it. Now we are, but we haven't had the need to mandate it yet because there's enough, we did surveys too. And so much of what we heard early on was, okay, thank you for all the education and everything you're doing, but how do I know I don't have it? How do I know I'm not asymptomatic? Or how do I know Keith next to me doesn't have it? So when the testing came, it was a big kind of sigh of relief. And, and as we turn our focus now onto the next phase, everything we just heard from the panel here is that there's still so much we don't know. So how much longer does a HABCO or any of the other businesses that out there have to deal with these protocols? So much of what we put in place is, a hit to efficiencies in some areas. So the testing allowed us, when we sent everybody off site on March 16th, we said, and when we forced people to go to second shift, we said, let's do it for 2 weeks and see how it goes. And here we are in week 14 now. And there's been a significant impact to people's home life. So now we could say, okay, if you want to switch to first shift, fine. We'll take a test. You get your negative result, come back to first shift. So we've been able to do a lot of, be a little bit more flexible with still never wavering on those 3 principles in the beginning. And now our focus is what's next, because it looks like for the foreseeable future, we still have to do this, right? The amount of time we spend every day taking temperatures at the beginning of each shift, the amount of time we spend cleaning at the beginning of shift, cleaning in the middle of the shift, cleaning between shifts, cleaning at second shift. So
are you looking for that?
I'm sorry?
Are you looking at new technology for those?
Yeah. So the 2 things in particular we're doing right now, we're going to start to look at a temperature taking kiosk. So just to automate that procedure, because I think we're going to be doing this for quite some time. And Carol and I have even had discussions, even post pandemic, come flu season and everything else. It'd be a good procedure to have. So it's a facial recognition kiosk where our employees can just walk by, take a temperature, could tell whether or not they have a mask on, if we need to have a mask before they enter the facility. So that's one thing. And then the other one that I'm really excited about is from a cleaning point of view and from how do we attack maybe the particulate matters and some of the whatever's in the air. So we're partnering with a company, our energy service provider who we get our LED lights from, and they're partnered with a company called ThinkLight, where they developed an air monitor. So we're going to be the first company, as far as we know, we'll be the first company in the country to put this air monitoring system in place where it can measure the particulate matters in VOCs, temperature, humidity. In our office, we can get some analytics. We can have a big and on showing, okay, green, our air is perfect, or hey, red, we got a problem. But what I really love about it is the ability to partner with this company, Think Light, with this product, and with World Energy, where we can spray titanium dioxide in the facility. There's a lot of information about titanium dioxide external use when it interacts with the sun, but this company has developed it where we can spray once every 3 to 5 years in our areas, and then the UVA emitters in our LED lights can interact with the titanium dioxide and look to kill a lot of the virus, bacteria, particulate matter, VOCs that are in the air. So to me, this is just a really good thing. We're still trying to learn a lot more about it. We've said yes that we'll move forward with it because, pandemic, non-pandemic, I would love to have better air quality and just a way for our employees to feel safe when they come in the facility to say, okay, your air is clean.
Yeah, that's super interesting. And so we just have a few minutes left, and I want to be respectful of everybody's time. But Brian, just picking up on that, and I just sort of want to quickly go through each of our panelists on this question. So, and I think that's sort of the number one general question we're getting is, what's the post-COVID-19 world going to look like? So assuming at some point there is a safe and effective vaccine, everybody has it, and we're back to normal, will we be back to normal? Are there things that have changed fundamentally for the good, for the bad, because of what we've learned and what we've experienced with COVID-19? So Brian, why don't you start with your business?
Yeah, it's interesting because Carol and I and our executive team, we talk about this quite a bit. There are some things that I don't ever want to change and go back to. One of the things we've realized is the amount of meetings we had before that we don't have meetings on now, how much productivity gain we have, right? By the time you go into a conference room, have a cup of coffee, start your meeting, it takes a lot longer. Now we hop on a Zoom call, we hop on a GoToMeeting, or we just pick up the phone and have that conversation. So we've had a lot of discussions about just the productivity gain of not needing to have a bunch of meetings. But I think what we've learned a lot on here is when we look at our productivity numbers during flu season and these other periods of time, when somebody gets sick and then you've got an outbreak within the facility, I think there's a lot of what we're doing now that we want to continue to keep in place. And like I said earlier, everything we're seeing and the unknown is still too great that we've got a long time before we're in a position to say we're back to normal, because I don't think there's ever going to be that normal again.
Sure. Clem, do you want to just talk quickly about what do you think the post-COVID-19 world will look like? Both maybe in scientific research or maybe at Sanofi or just...
I think a lot of Brian's comments about meetings and things like that, I think, are expected to change. The other thing is, I think some of the lessons learned from vaccine development here will be applied and will accelerate vaccine development. And the third thing, but I do think that's going to decline, is I think we are now acutely aware of the potential impact of a pandemic, and we might be more willing to invest in pandemic preparedness. But as the memory of COVID-19 fades, I'm concerned that we'll go back to where we were before. I think this highlights the need to be prepared to respond. And, I commend, particularly Carol and Brian, because it's fascinating to hear what they've done and how they adapted, and those lessons learned would help them respond to another emergency, and also share those lessons with people, because it's, as you are doing today, because it's fascinating.
Thanks.
Great point. Charles, so tell us about the post-COVID-19 world from your perspective.
I would echo, especially the last thing that Clement said. I think we're going to be a lot prepared for the next wave of pandemic. There will be another wave of something that will go through. And I think from the experiences we have right now, whether it's with respect to having enough PPEs, able to respond quickly to testing, to be able to, provide, make sure we have enough infrastructure for vaccine developments, et cetera. I'd like to be optimistic and think that when the next pandemic comes through, we'll be a lot more prepared on all of these levels.
It's a great note to end on. I want to thank all of you, all of our panelists, so much for your time and your insight and your wisdom about COVID-19 and the novel coronavirus. And thank you, all of you tuning in to the webinar, all the CBIA members. Thank you so much for being part of it. So with that, we'll close this out. And again, it will be on the CBI. This webinar will be on the CBI website. Thank you so much.